Hi, my name is Deeksha Satish, and my project was on the discovery and analysis of a novel microbacteriophage. So my research was done as a part of the phages program that is run by the Howard Hughes Medical Institute. So essentially they provided us with all of the procedures so there'd be a consistency throughout the program. And so why study bacteriophages? Some estimates say there are 10 to 31 bacteriophage species present on this planet, making them the largest biological subsection. Yet we have barely made a dent in characterizing their genome and understanding their protein functions. So my research will help add to the scientific community's knowledge of bacteriophages. And there are some real-world implications of this as well. One application is phage therapy and replacement of antibiotics. As antibiotic resistance continues to grow in bacteria, phage therapy has been showing promise in combating such infections in humans. So the first step of this process was isolation. I took my environmental sample from a compost pile in Bowling Green, Kentucky, and this is because the bacterial host cell, Microbacterium foliorum, is known to grow around decaying plant matter. So naturally the phage that targets the host will be found in the same general area. From here, we enriched the sample with host cells and calcium chloride, and then plated it on a host bacterial lawn and looked for any plaques, which indicates a phage present. From here, we moved into purification. Essentially, we only want to be studying one phage at a time. And what we're looking for in the purification process is a consistent morphology on our plates. So as you can see on the second serial dilution, all my plaques look the same. So that indicates to us that we only have one phage species present. In addition, we also look at the concentration of plaque forming units per milliliter. And the formula for that is number of plaques counted on the plate over the volume plated times the dilution factor. And for the second serial dilution, we calculated 9 times 10 to the 6th PFU per mil. From here, what we do is we fled this plate with a phage buffer to suspend the phages in it. And from there, we can use that mixed with host bacterial cells to make 10 such plates. And this essentially allows the bacteria to replicate and for us to get a high concentration solution of phage. This is a quick look at the plaque morphology. After 48 hours of incubation, the plaques had a diameter of 1.5 millimeters. In addition, as you can see, they are very cloudy or turbid, and turbidity essentially indicates a lysogenic life cycle, which is where the phage integrates into the genome and waits for optimal conditions before entering the lytic life cycle. So that's why the plaques appear turbid. It's because not all of the bacteria have been killed inside of them. And so essentially what we did after we purified was we looked into the genomic characteristics and the physical characteristics as well. First, what we had to do for gel electrophoresis was extract DNA. And this is where the high concentration solution comes into play. Since phage DNA is very small, we need a high volume of it to be able to get a good, clear gel electrophoresis picture. So what we first had to do was extract the DNA from the phage. And to do that, we essentially just broke down the protein surrounding the DNA and then extracted it through a filter. And then what we did is we ran a restriction digest, which is essentially where we mixed the DNA samples with 5 different restriction enzymes. And what this allows us to do is test for 5 different restriction sites. As you can see, this is our gel electrophoresis picture. This is our uncut DNA, it's the control. 4 of the restriction enzymes remained at the same level as the uncut DNA, and what this tells us is that there are not many, if any, restriction sites for these 4 enzymes on the genome. Now for WEL6, which corresponds with the HAE3 enzyme, there is a smear. What this indicates is that there are many restriction sites for that enzyme. And so the Hay3 restriction site is GGCC, which means there are many GGCC sequences on our genome. So this gives us just a general understanding of what our genome looks like. So going into electron microscopy, Squeegee, which was the name of my phage, had an icosahedral head and a cyphaloidal tail. The tail is long and slightly curved. As you can see, it also exhibits a characteristic called sticky heads, which is essentially where the heads tend to clump together. We were also able to measure the average capsid diameter and the average tail length, 43.9 nanometers and the 130.4 nanometers respectively. So at the end of all of this, here are just the results. So we were able to conclude that Squeegee had a GGCC rich genome. It's a temperate phage because it goes through the lytic and the lysogenic life cycle. Its physical characteristics are that it has an icosahedral head and a cyphaloidal tail. And at the end of all of this, we were able to upload this data from my phage onto the database PhagesDB. And so Dr. Rodney King was my research mentor through all of this, and Dr. John Andersland helped us with the electron microscopy part of it. Here are my works cited. Thank you for listening.